Welcome, new USU medical students. My name is Stephen Davies. I'm a veterinarian and an infectious disease specialist in the Department of Preventive Medicine and Biostatistics. And I'm here to give you a brief introduction to the topic of immunology. Immunology is a foundational subject in human medicine, so it's a topic we'll be covering in some detail soon after you arrive at USU this fall. So what is immunology? While immunology is relevant in many areas in human medicine, it's easiest to introduce in the context of infectious diseases. We all experience infections from time to time. Perhaps you had an upper respiratory infection recently, a common cold perhaps, with mucus production, sneezing, etc. Or perhaps you experienced something more serious that resulted in you running a fever for a few days, something like influenza, or maybe COVID. Or perhaps you experienced gastrointestinal symptoms, something we'd call a stomach bug. Whatever the symptoms, the reason you experience them is because you are infected with a pathogen. This is usually a virus or perhaps a bacterium, but sometimes other pathogens are involved, such as fungi and parasites. However, with time, your symptoms abated and you gradually returned to normal. Your recovery from this infection was due in no small part to the fact that your body mounted a response against the infection, a response called an immune response, a response that led to the control of the infection and ultimately the elimination of the pathogen from your body. These immune responses are mounted by a specialized system of cells and tissues called the immune system that has specifically evolved to protect us from infections. This is what immunology is. It's the study of the immune system and of the immune responses that it mounts in order to understand how these impact human health and disease. I mentioned that infections can be caused by a variety of different kinds of pathogen, everything from viruses to bacteria to fungi and parasites, which can even include very complex multicellular organisms such as parasitic worms. You'll learn more about the diversity of pathogens that can cause infectious diseases in humans elsewhere in your curriculum. Think of the immune system as your body's defense against these myriad infectious threats. It's essentially your body's Department of Defense, with assets deployed throughout the body to prevent pathogens from gaining a foothold and to eliminate those pathogens that do manage to establish an infection. One of the most important applications of immunology in human medicine has, of course, been the harnessing of the defensive anti-pathogen properties of immune responses through the development of vaccines. Some infections, diseases like measles, for example, induce immune responses that provide long-lasting protection, sometimes lifelong protection, against any further infections with that same pathogen, and generally prevent you from ever experiencing that infection again. Vaccines are designed to induce these long-lasting protective immune responses without the need for that initial infection. Indeed, vaccination has been so successful that it is now one of our most important tools for controlling some infectious diseases, both for individual patients and at the population level. It will be important for you to understand the immunology behind how vaccines work so that you can give your future patients clear, evidence-based advice on how vaccines might be relevant to their health. In fact, some infectious diseases are no longer a concern for human health, thanks to their effective control through vaccination. Take smallpox, for example. This is a terrifying infection caused by the smallpox virus. Smallpox has a 30% fatality rate and killed around 300 million people over the course of the 20th century alone. That is, until it was eradicated from the planet in the late 1970s by a worldwide vaccination campaign orchestrated by the World Health Organization. The last naturally occurring smallpox infection was recorded in Somalia in 1977, And after 2 years where no new cases were detected, the WHO was able to officially declare that smallpox had been eradicated in 1979. This is a truly remarkable accomplishment that has averted an untold amount of human suffering. We've talked about how immunology is important in the context of infectious disease, but as you will learn, immunology has relevance throughout human medicine. Here are just a few other examples. Interested in reproductive medicine, perhaps? There's a delicate immunological interplay that occurs between a pregnant woman and her developing fetus that has important implications for the health of the mother and her baby. Want to be a transplant surgeon? It's not all about cutting things up and sewing them back together. You'll need to also consider the immunological interactions that occur between the grafted tissue and the recipient's immune system to achieve a successful outcome for your patient. And immune responses can cause disease when they are directed towards inappropriate targets. Take allergies, for example, which occur when immune responses develop towards innocuous environmental substances. This is just one example, the rash caused by exposure to the sap of poison ivy, which gets into the skin and induces an inappropriate immune response. I'm sure some of you have first-hand experience of this. And immune responses can be the cause of very serious diseases when they target components of our own bodies. These are diseases we call autoimmune diseases. Rheumatoid arthritis, characterized by severe joint pathology, is but one example of an autoimmune disease. Now that I've convinced you of how important and relevant the immune system is, I'd like to spend the next couple of slides introducing you to the composition and organization of the immune system. Just like other organ systems, like the nervous system, the digestive system, and so on, the immune system is composed of specialized cells that are organized into tissues and organs. Interestingly, the cells of the immune system all develop from the same stem cells in the bone marrow that give rise to red blood cells and platelets. In fact, many immune cells circulate in the blood with the red cells until they are called upon to participate in immune responses. These are the white blood cells, such as monocytes and neutrophils. On the other hand, other immune cells, such as mast cells, transit only briefly through the blood on their way from the bone marrow to other tissues where they perform their functions. In addition to the blood, immune cells are also found in specialized tissues of the immune system that are referred to as lymphoid tissues. We recognize two functionally distinct types of lymphoid tissue. There are primary lymphoid tissues, in red, such as the bone marrow, which are sites where immune cells develop. And there are secondary lymphoid tissues, shown in yellow, which are mostly lymph nodes, where immune cells go to participate in immune responses. Lastly, I'd like to briefly introduce you to some functional aspects of immune responses themselves. So there are two main types of immune response. The first, and most intuitively obvious, are the types of immune responses we've already talked about. Provide long-lasting protection against specific infectious diseases. These are the type of immune responses that are also induced by vaccines. These are called adaptive immune responses. These responses are highly specific, targeting specific pathogens, but they take time to develop, usually on the order of a couple of weeks. These adaptive immune responses are mounted by specific types of small lymphocyte, a type of white blood cell, called B lymphocytes and T lymphocytes, or simply B cells and T cells for short. The second type of immune responses are called innate immune responses. These responses do not provide long-lasting protection and are not specific to certain pathogens, but they are nonetheless very important because they develop rapidly, beginning as soon as a pathogen enters the body. Innate responses are mediated by different cells to adaptive responses involving various cells such as neutrophils and macrophages. The reason innate responses are less specific is because innate immune cells recognize pathogens using receptors for molecules that are shared by broad groups of pathogens. A carbohydrate, for example, that's shared by a group of similar bacteria. That carbohydrate receptor confers on the cell the ability to recognize that group of bacteria. These receptors are called pattern recognition receptors, or PRRs, and the pathogen molecules they recognize are called pathogen-associated molecular patterns, or PAMPs. Each innate immune cell will express a small repertoire of pattern recognition receptors that collectively give it the ability to recognize a variety of different pathogens. Pattern recognition receptors allow innate immune cells to physically interact with pathogens through their PAMPs. As many innate immune cells have phagocytic capabilities, this interaction leads to the phagocytosis of the pathogen, i.e., it is taken up into the cell in a membrane-bound vesicle called a phagosome. This phagosome will then fuse with another type of vesicle called a lysosome, forming a phagolysosome. This exposes the pathogen to the contents of the lysosome, which includes proteolytic enzymes and other substances that are toxic to the pathogen, thus resulting in the death and degradation of the pathogen. This is how innate immune responses often contribute to defense against infections by eliminating the invading pathogens through phagocytosis. On the other hand, the B lymphocytes and T lymphocytes that mediate adaptive immune responses recognize pathogens by a fundamentally different mechanism. Instead of expressing a repertoire of pattern recognition receptors, every B cell and T cell expresses just a single receptor with a single specificity. Furthermore, that receptor and its specificity is unique to that cell, such that almost no two lymphocytes possess the same specificity. This means that in a human body that contains billions of lymphocytes, those lymphocytes collectively are able to recognize billions of different foreign molecules. This enormous diversity of specificities ensures that whenever a pathogen enters the body, there will always be at least some lymphocytes that are able to recognize molecules from that pathogen. The downside of this approach is that on first encounter, there will only ever be a small number of cells that can recognize that pathogen, and those cells need to multiply through proliferation until there is enough of them to mediate an immune response. This explains why adaptive immune responses take on the order over a week or so to become effective. Once the adaptive immune response is in full swing, B lymphocytes and T lymphocytes contribute to host defense in different ways. B cells secrete proteins called antibodies, which are simply soluble versions of the same membrane-bound receptor that the B cell used to recognize the pathogen in the first place. The antibodies bind to the pathogen, inhibiting its cellular functions and targeting it for destruction. T lymphocytes, on the other hand, contribute to defense by two different mechanisms. Some T cells interact and stimulate other immune cells to promote their defensive functions. Some T cells will interact with macrophages, for example, and stimulate their phagocytic capabilities. Other cells will interact with B cells to improve their antibody production. These T cells are called helper T cells in recognition of the role they play in helping other immune cells. Other T cells play a specific role in controlling virus infections by killing cells already infected with virus, thereby preventing them from becoming a source of additional virus that could spread to other cells. These T cells are called cytotoxic T cells in recognition of their cell-killing capabilities. The reason adaptive immune responses are able to generate long-lasting protection against pathogens is because at the end of an adaptive response, once the pathogen has been cleared, there's a phase when special types of B cells and T cells develop, called memory B cells and memory T cells. These cells are long-lived, and as their name suggests, they retain the ability to recognize the original pathogen and respond to it much more quickly if the pathogen ever enters the body again. It's these memory cells that are responsible for what's called immunological memory. Thus, in this example, at the end of the first adaptive response, which might have been induced by an actual infection or a vaccine, long-lived memory cells were formed. Then, when this pathogen entered the body again, at a later date, sometimes years later, those memory cells are still present. They recognize the pathogen and responded to it very quickly. This secondary response can be so quick and effective that it prevents the infection from becoming established, thus preventing the development of any disease or symptoms. Last of all, I've presented innate and adaptive immune responses as two separate processes, but in reality they work hand in hand with each other, contributing to an effective overall defense in different ways. While adaptive immune responses provide more specific, long-lasting protection, this takes time to develop. Innate immune responses, on the other hand, develop quickly and provide a critical first line of defense while the adaptive response is still developing. Okay, I hope this short introduction has piqued your interest in immunology. I look forward to exploring this fascinating topic with you in greater detail when you join us here at USU this fall. I'll leave you with this image of Ali Ma'aleen, a Somali hospital cook and healthcare worker who was the last person to be naturally infected with smallpox in 1977 when he was 23 years old. He went on to make a full recovery and later dedicated his life to improving the health of his community through vaccine campaigns for other diseases. Thank you for your attention. If you have questions or comments, please don't hesitate to contact me.